on how to work in the terminal, how to use command line interface, less about the NGS analysis in the first part. We will touch upon that in the workshop. So yeah, firstly, I would like to thank my colleague, Ivar Silmichels, for helping me prepare this presentation. Yeah, so bioinformatics, it's an interdisciplinary field which combines statistics, biology, and computer science. In my opinion, there are 3 main ways in which you can become a bioinformatician. If you are a student of biology, like me, then you have to learn statistics and some programming. In my opinion, that's the hardest way to go about it. If you're a statistician, then you need to learn something about biology, and you probably already know a lot about programming or computer science, because those fields often are very closely related. Or if you are a computer science person, then the same as for the statistician, you need to learn a little bit of biology, but you most probably already know quite a lot about statistics. But I want to mention that you don't need to become a bioinformatician or call yourself that to analyze data, because there are many different levels to this, and I like to think that there are 2 types of people who analyze data, who do bioinformatics basically. So one is a person with a computer science or statistics background, and they most often than not write software for bioinformatics. Because that's, in my opinion, that's the difficult part, or the more difficult part. Or you are the person who mainly does data analysis and result interpretation. That's something along the lines of what I do. Yeah, so these are 3 of the most common operating systems used in bioinformatics. Actually, I'm not sure about the FreeBSD. Haven't heard that much about it. I know that the macOS is quite popular because it has similar roots to Linux. They're both based on the Unix operating system, and it comes preloaded with a Linux type terminal. So and a lot of the commands are very similar, and you don't need to do anything to set it up, basically. And of course, there's Linux, but when I say Linux, there's no such thing as Linux, the operating system. Basically, Linux is a base, and then you have many different distributions or flavors of Linux. For example, when Laura sent out the setting up the working environment PDF, I asked you to install Ubuntu, and that's one flavor of the Linux ecosystem. So yeah, you can choose basically any Linux distribution. Doesn't matter that much. They have differences, of course. Also, they have a graphical interface just like Windows or macOS, but most of the programs for bioinformatics are made without a graphical user interface. So that's why most often than not we use terminal. Okay, yeah. So why Linux? Because the best software for bioinformatics is written for Linux. It's easy to develop new tools. Of course, that's subjective. What is easy for who? But relatively I think, a lot easier than creating software for Windows or other operating systems. Easy to implement simple workflows because we can write bash scripts, which I will tell you about later. And you can combine those bash scripts, and there are other specific programs which allow you to create a simple but very effective and fast workflows. Good reproducibility, that means that basically we can track everything that we are doing with relative ease. We can know where the data is coming from, where is it going, what software was used, what is the versions of the software, which databases, versions of the databases, and so on. So we can further pass that information to other people who want to reproduce our work or for publications. So the Linux-based operating systems are commonly used in high-performance computing, and that goes together well with, of course, the operating systems that we are using for data analysis, which is also Linux-based. So the packages often are the same, and they are made to have good integration, and they can work together very efficiently. So yeah, and you can modify anything you want, of course, if you know how to, and if you're brave enough to do it, because often the source code for bioinformatics software is available. You can read it, of course, if you know how to, and you can modify it and compile the program again. Sometimes it's actually necessary, because if you want to be on the cutting edge, sort of, you sometimes run into problems when a bioinformatics software is written, but it's not well maintained, and it's quite a big problem actually in bioinformatics. That there's a lot of software. It's produced in large quantities, but the maintaining of it and documentation is not always there. And that also goes for the support when you have to contact the developer if you want something from him, help or anything. That's not always possible. And then, of course, it's free. You don't have to pay for anything, not for updates. You don't have to buy it, and so. Yeah. So Ubuntu. I think it's the most popular Linux distribution, developed by Canonical. Based on Debian. I think it's very user-friendly, not from the perspective of which we'll be using it, maybe. If you never worked on a terminal before, then it's maybe not so user-friendly, but it has a graphical user interface, and you can easily replace your Windows operating system with Ubuntu, and there won't be much difference, in my opinion. Yeah, it's easy to install, and and you can install the Ubuntu subsystem for Windows 10, Windows 11, just like I wrote in the tutorial for setting up the workplace environment. So this is the graphical user interface of Ubuntu. In the background, you can see the terminal. So somebody is connected to it. There are all the files and directories, and in the front, we can see the actual typical window of a folder. So it's very similar to Windows, in my opinion. Yeah, so here's a terminal. Really, there's nothing much to it. It's the same as running any other software graphically, just we don't have the pretty colors maybe, and the ability to click with our mouse. But it's definitely a lot more efficient when you have to deal with a lot of data, and in genomics and omics data analysis, there usually is a lot of data. It takes up a lot of space, and you need a lot of resources to compute it. So terminal is definitely the way to go. Yeah, so In Linux, everything is a file or a directory. Yeah, each file has a location in the system, also called path. So there are two types of paths: relative paths, which basically start from the working directory, or you can think of the working directory as the directory in which you are currently located in, or there are absolute paths starting from the root directory, which means that you have to basically type out the full path to the directory or the file that you want to operate in or on. So each directory is separated by the /, and here's an example of a path. So this will, this will be a relative path because we can see that there is no root directory in front of it. So basically we are already in some directory, and we want to, let's say, access this file.txt, which is located in two other nested directories. Root directory is /, one directory up, or maybe I don't know if up is the best way to say it, but I think the better way to say it is backwards. So if you want to go back one directory, then it's 2 points. If you want to specify the current directory, then that's 1 point. And the user's home directory is the tilde sign, which is basically another way to look at it is if we have the root directory, it's usually root, home, and for example, Rihards. That will be my home directory. And everything else that I'm working on is probably based out of that home directory. So here are some examples of navigating the directory system. For example, if we are located in the home directory and we want to specify the user directory, then there are two ways. Of course, home user, that will be the full or absolute path, or the relative path would be just user. This will make more sense a little later when we will look at the commands which allow us to move through directories. So when we add the command and the full path or the relative path, it will make more sense. But basically, if we look at the structure, so we're starting from here, and just relative path would be just one folder forward. An absolute path would be home, root, home, and then user. So similar with if we want to specify the documents directory, it would be home, root, home, user, and documents, or just user and documents, because this is our working directory and we start from here. That will be the relative path. And similar with the file data.txt, we can specify the full path or the relative path, but I guess it's always better to write less if you know shortcuts, or if you know the fastest way. It's always I think it's always better to use the shortest way or the fastest way. Also, if you type less, there's less chance that you'll make errors, which from my experience from typing directories often and moving through them, it's very annoying. You mistype the names, and if you can do it in a shorter way, that's probably the best way to do it. Also, there's need to be taken into consideration the fact that it might be good if we name the directories in some consistent manner. Let's say if we have a directory starting with lower caps like home, user, those are the default. We don't make those, but let's say we want to make the directory documents. So maybe it would be better to make it with a lower D, not the upper D, because if you have to type it many times over and over and over, the pressing the caps button and then the D or the shift D, that's like an extra step in which you can make a mistake, and over time it does get very annoying. So following a naming convention is not a bad thing to do. So yeah, there's another example. If your working directory is user and we want to get to the data.txt file, again, very similar to before, we just moved up one directory. Yeah, so if we're in documents, and obviously the file data.txt is in the documents directory because there is no other directory in between us. So the relative path to that document will be just data.txt. Or if you want the full path, then it's all the way from root to the file. Sometimes we are often when I work in this kind of environment, I make multiple subdirectories where they branch off, and let's say I have some sort of database or program stored in another directory, which if you look at the path, it's backwards. We need to go back to access it. So one way to do it would be to, of course, type the full path just to that directory or file or software, whatever. Or we could just use the relative path by, as I mentioned before, 2 dots means that we are going one directory up. If we would want to go 2 directories back, we would do 2 dots / 2 dots. So yeah, let's say we want to go to the data.txt, we are in desktop, so we can just go 2 dots. So we're back in the user, and then user documents data.txt. That's I think that's more efficient and a better way to do it than just type the full path, which in some cases the full path can be absurdly long, and also depending on how long are the names of the directories that you made or that were already there. So the relative paths are suggested. Yeah, let's say we want to specify the same directory that we're already in. So just the relative path to home from home would be just a single dot. Sometimes when you're running some sort of software and it asks you to specify where you want the output to go, and if you want the output to go in the same folder that you're running the software from, or that you're in right now, then you just specify the dot and it will write it there. Of course, you could also write the full path, but if you can just use the dot, then why do it? Yeah, so the file and directory naming conventions, these are all the things that you can use. I would suggest for ease of use, probably to use all lower caps. If we want the name of the directory to be multiple words, I know it's like a bad habit from Windows to separate those words with spaces, but spaces can create problems, or they just complicate things when you have to provide the path using that directory. So it's better to use underscore or dot in this case. Also, probably the best way is not to mix, not to mix lower and uppercase, just for the sake of simplicity. As I mentioned before, pressing the all caps, making mistakes, and so on. Yeah, and also don't begin the file names with the dash. And files that start with the dot, they are hidden. There usually are several hidden files, especially, let's say, in Ubuntu. When we log on to a server, there's a file called bashrc, and another file called bash profile, and usually we can't see them. They are available from any directory. So when we want to open those files, we have to put the dot before the, let's say, bash rc or bash profile, and those files are usually used to set up some kind of environment variables, or let's say we can provide the directories for specific software. So when we want to use that software, we don't have to type the full path. We can just type the name of the software, and by providing those directories and those files, it will know where to look for that software. So here we have a list of a couple, I'd say, most popular commands used in the bash shell. So we will go through them in more detail in the next slides. Maybe not all of them, and I think that some are missing from here. Maybe I'll show them when we have the workshop. Yeah. So there's the anatomy, basically, of using the commands. If we have some kind of default commands on Linux or through the bash shell. So the bash shell is basically sort of like a program in the Linux that allows us to run and interpret these commands. We'll touch on that later also. So first we have to type, let's say, ls, which means list. Then we type the options that we want to use with this command. So for each program, basically the ls command or list command, it's a program. Each of these commands is a program on its own, and it can have options or arguments. So they are often preceded with a single slash or double slash. Yeah. So, and after providing those options, we can, let's say, provide the file or whatever else the input is for that command. If that's a default command, let's say on Ubuntu, there are often available manuals for us to read on how to use it, and we can access those manuals by typing the command man and the name of the command for which we want to see the manual of. That's also very useful, especially for beginners, and not only for beginners, because obviously you can't remember anything with time. After using these commands more and more, we'll start to remember it, but we can't remember all the options. And usually there are a lot of options for these commands, and each of them do something different. And more often than not, there are some edge cases or specific cases where we need an option or a combination of options, and therefore we can look it up using the man command. So yeah, list. So there are some commonly used options. If we type ls dash a, then we'll see all the files in the directory, all the files that we would normally see by typing ls, and also the hidden files, as I mentioned before, which start with the dot. Use a -l, use a long listing format. Basically, it means that it gives you more information than just the file name. It gives you, I think, the size of the file in bytes, the date it was created, and the permission for this file, like who can read it, who can write in it, and so on. If we combine ls with -l and h, so yeah, actually, if we want to, let's say we want to combine L and H options. So we would type ls-lh. We could also type ls-l space -h, but it's not necessary if we can just type them with a single dash. And let's say we combine the L and H, and we would get sizes of all the listed files in a readable format, which in my that's like kilobytes, megabytes, and gigabytes. Of course, bytes is not very easy to understand for an everyday user, and I don't think anyone wants to do the hassle with trying to figure out how much is I don't know, 1,000 bytes or whatever in megabytes or something. Yeah, so -t, we can sort the files by modification time. -r, reverse the order, and -d, list the directories, not the contents. So cd, or change working directory. So ls and cd, those are the commands which you will most likely use most often when working in a command line interface. Sometimes when you are thinking about something, or just, I don't know, you're bored, you would just type ls in the terminal for no reason, out of habit, for minutes, many minutes straight. So yeah, just with time you start to remember them, and yeah, to the cd. So cd command allows us to change the working directory, which goes together with what I was talking about before, about the directory system and how to move through it. So let's say we want to go to the directory documents, we just type cd documents. If we want to go to the home directory, cd tilde, same as cd nothing, one directory back, cd space dot dot. This is actually a very useful option, of which I learned not that long ago, actually, after doing this for 4 years. So sometimes when you're typing directories and you mistype it and end up in the wrong directory, or you forgot where you were before, which can happen, you can just type cd and dash, and it will take you back to the previous directory, regardless of how far away or where in the file system it was. And yeah, these two options I haven't used, -l and -p. Most likely because they are for some specific edge cases. mkdir, or make a new directory, which we will be doing this in the workshop. Common used options, -p allows us to make several nesting directories. For example, if let's say we want to make a directory results, and in the results directory we want to make another directory, for example test. We don't have to type the command mkdir twice. We can just type mkdir results /test, and it will create both of the directories in one command. And if we provide the -v option, then for every directory created, it will print a message on the terminal that it has created this directory. So copy file allows us to copy files from one directory to another. Also, it can copy directories itself, but we need to provide the -r option. If we provide the -i option, then it will ask us if we want to proceed with this action. -s allows us to make symbolic links, which basically is if we have files in one location and we don't necessarily want to copy them to another location, but we want to have access to them, we can create some symbolic links, which is sort of it's sort of like a hyperlink. When you have a hyperlink in a, let's say, in a Word file, and you click on it and it takes you to a web page. So in this case, we have the symbolic link for a file, and it's like it's not the file itself, but it knows where the file is from that symbolic link, and we can still work on these files and use them, read them, and so on. So the next command is move. In my experience, this is a very useful. Of course, we can move files with this, but in my case, the most times I've used it is actually to rename files, not to move them. Which is, I guess, technically it's still moving them because we are moving them from a directory with one name to a directory of with another name, but we are not saving the previous directory. So let's say I make a directory test1, but I actually wanted to call it a test, so I'll just type move test1 space test, and it will create the test directory with all its contents and remove the test1 directory. If we add the -n option, of course, it will not delete the previous directory or file. And -v again, -v often stands for verbose, which means that whatever is happening will be printed out on the terminal, so you can follow the progress in, let's say, in real time. Remove, you have to be careful with this one because it's possible to delete everything. It can remove files. It can remove directories if we provide the -r option. If we, let's say, sometimes some files have different permissions and we can't delete them simply by typing the remove command, or the same goes for directories, but we can type remove -rf, on which will the f option will force it to be deleted. The rest of the options are basically similar as before. So here's a meme. As I told you before, the / just the / is the root directory, and all of the file system is based from the root directory. So if we sudo rm -rf and the root, we will delete everything in the file system and we will have to reinstall the operating system. So do be very careful. But as you can see here, there's a command called sudo in front of the rm or remove. So the sudo is basically like on Windows, when you don't have the administrative privileges, but you need to install something or delete something, you need those privileges, and the sudo is the way to access them. There's a basically sudo means like super user do. So super user means we have all the permissions most often than not, and with most likely without the sudo we can't delete anything. It will not be permitted. But if we can use the sudo and enter the password for the sudo profile, then we can delete everything by accident, let's say. So less, this is a very useful command. I use it very often. Basically, when we have let's say, a very large file, which is often the case when we are working with genomics data, those files can be in many gigabytes or often larger, even hundreds. So normally on Windows, when we want to look at a file, let's say we want to look at what's inside the file, but we only want to look at the first 5 lines or something. To do that, we would have to, let's say, open it in Notepad or Word, but it's not realistic to read the whole file in the memory. So when we want to open a file in Word or Notepad, the file has to be read into our in memory or RAM, operating memory, and it's a very limited resource, often not in large quantities. And even if you manage to do it, if you have enough RAM, it will most likely freeze your computer. So a good alternative is to use the less command. It just basically opens the file line by line. It doesn't read it into the memory, so it's very fast. it's efficient, and you can also search the file by using the less command. Very useful option for this command is -s, which is not written here, but -s means that often, let's say, we're working with some sequencing data and we have very long sequences. So normally, when we open the file with less, it would wrap the line. So it would transfer the part that doesn't fit in our screen, it will transfer to the next line. And when we have a lot of similar looking data in the file, it becomes a mess on the screen and it's hard to understand what's going on. So when we type less-s, it disables the line wrapping, and you actually have to move to the right to read the rest of the line, but the file itself becomes more readable. There are some options which allow us to quickly move through the file. If we're opening a file with less and we have gone to, I don't know, the 1000 line, and we quickly want to go to the first line, we can just double tap GG on our keyboard and it will go to the first line, or Shift G, go to the last line, and or -N, it will show us the line numbers. So cat, actually use this command quite a bit, because sometimes when we have files that are coming from sequencing platforms, sometimes some samples are resequenced multiple times. So before analysis, they have to be combined. And if they're in a Fastq format or gun-zipped or just zipped Fastq format, basically, we can concatenate those files easily and create one file containing sequences from both of those files. But that's those sequences from two files, but that's just one sample, and that's often the case, and therefore this command is very useful. And yeah. Head allows us to print first n lines. I know it's written 10 lines, but you can specify the number of lines. Let's say n1 or n,000, and it will output to the terminal those first n lines that you specify. So there's a mistake. There should be a command tail, not head, and this command will print the last n lines, not the first n lines. Options the same as before. These are useful when you have some sort of sorted files. Let's say they're sorted by chromosomes, or I don't know, they're sorted by sample identifiers, which often contain numbers. So you can quickly look at the last samples, the first samples, and so on. So WC. For me, I use this command to check how many lines are in a file. If you type WC space -L, it will output a number of lines of a file. And let's say we can use it also to, in combination with a different command called BC, it's also in Bash. We can combine those two commands and, let's say, calculate how many reads are in a sequencing output file. We need the BC command because actually each read consists of 4 lines, so we need to divide the line count by 4. We'll talk about Fastq files later. Cut allows us to select fields from a file. So by fields, I mean columns. Let's say we want only the first 5 columns or something. We can specify cut space -F1 -5 or -F1, -2, -3, -4, -5. Or yeah, we can also specify specific delimiters. Delimiters are the invisible symbols which separate our columns. It could be tab, it could be space, it could be also a comma, a pipe, whatever. So we can specify the delimiter that we want to, and it also allows us to manipulate the data. But so because often not all file formats are consistent in their delimiters. We could have like the first 5 columns separated by tab, and then we could have a last column which includes a lot of information, and the information in that column is for each field is separated by a pipe, for example. That is the case in the VCF file that we will look at later. So echo, this is a command that we can use to print output to the terminal. I often use this for debugging or to write something to a file. Let's say I'm creating a table from some quality check output of sequencing reads, or as I said, debugging. I mean, I can check, I can write the script, but I don't write the script as a whole in one go. I can write a part of the script, and I can echo the some output from the first part to make sure that I'm on the right path, that I haven't made a mistake. Let's say that it finds the files that I want to input, and so on. Yeah, for example, if we want to print out a line which let's say with 3 words, and we want to separate those words with a tab, we would add the -e option, and that basically means that because everything in quotes is considered a string, and if we wouldn't add the option, it would just print the \t, which is a symbol for the tab. It would just print it as is. But if we add the -e option, it enables the interpretation of it as a separator symbol, not just characters. So yeah, file editors, there are many. We are going to be working a little bit with Nano today. It comes by default with Ubuntu. It's simple. I don't like it, but it does the job. And yeah, you can create files with it. You can edit files with it. I'm sure it's more powerful than I realized, but I haven't gotten into the habit of using it. And so, yeah. So the greater than sign, basically, when we run a command in the terminal, as mentioned before, it might require options. It requires input, let's say a file. And if that command modifies the file, we can specify it to save the output to another file. Or there's an example. If we want to concatenate 2 files, f1.txt and f2.txt, we provide them as input to the cat command, and we use the greater than sign to point the output to the f_concat.txt. The file doesn't have to exist. It will create the file if it doesn't exist. Yeah, here's another example using the cut. If we want to cut the first column or field from a file, you can just type cut-f1 for the first column, the input file, use the greater than sign to redirect the output to call 1.txt. So file name globbing. This is the sort of thing that if you end up working with some type of genomics data, you will encounter this a lot. It's super useful. because usually the genomics data comes from multiple samples. Let's say from a run, from an experiment. There usually are multiple samples, and you need to perform some sort of action on all of them. We could just retype our desired command for each of the file, but that's tedious. There's a big probability that we will make mistakes by typing them. And so this file name globbing, let's say with using the wildcard of star, allows us to create a list of files matching a pattern. In this example, we will create a list with the ls command, list of all files, let's say in a directory, which end with the txt extension. So basically this star means that select any and all the files. Doesn't matter what comes before the dot, as long as the files end with the txt extension. Yeah. So the question mark allows us to match one symbol. We can use multiple question marks. And in this example, we would select any and all files which have anything before the dot and that have 3 symbols in the file name extension. The brackets means that we are matching one, or if possible, multiple symbols, which we have specified in the brackets. So basically, if we are in the directory and we want to make a subset of files, and those file names have numbers before the dot, like file 5 or 510 or whatever, and we want to grab the files which have 1, 3, or 9. So the output of this will be file 1, file 3, and file 9.txt. The exclamation mark, not just in the terminal, not just in the bash. Often it means to do the opposite. It means to not do something. So using the same example as before, we don't want to select files which contain 1, 3, or 9 in their name. Square brackets, you could think of it like an and. It's not if you use the if you use the square brackets, then it's more like an or statement. If you use the curly brackets, then it's more like an and statement. We want files that are either that are txt and that are doc in the directory. So here are several few examples of this. So let's say we want to just list the file f1.txt, and we would get this file. That's very obvious. Here we want to select file which name starts with f, and the next symbol is anything, and the next symbol is dot. So yeah, it doesn't mean that we basically, this doesn't mean that there has to be exactly one symbol. For that we would use the question mark. This makes it so that there could be a million symbols or just one. It doesn't matter. So we would select all of these files. Here we select files which start with f and end with 0 before the dot, which would be the f10 and f100. Yeah, so f and one single value, doesn't matter which one, before the dot. There's only one kind of file with this kind of name, the f1. So the one was the value in the question mark. 3 values after f. Doesn't matter which values, it matters that there are 3 values. And that's the F100 file. F1 and 1 or 2, that's F11 and F12. F1 and not 1 or 2, that's F10. F and 111, that's F11 and F100, pretty self-explanatory. So here are the links to some resources about Linux, where you can learn something about this more extensively, of course. So this is a similar example as I told you about before, so I think we won't go over this again. Oh, actually the double greater than signs. So if one greater than sign means that we are saving the output to, let's say, a file, then 2 greater than signs means that we are appending to a file. If we already have an existing file and we want to append some information from a different file, we would use the double greater than sign. Let's say I have 2 files. I have the reference genomes for chromosome 1 and chromosome 2, and I want to combine them. So I would use I could append the information from the file of chromosome 2 to the information of the file of chromosome 1. So the pipe operator, also very useful. I will show you an example of it in the workshop. Basically what it does, so if we're running a command and we are not using the greater than sign to specify where's the output going, then the output is going to the standard output. That's what it's called. So it has programs have standard input and standard output, and the pipe operator allows us to direct the standard output from one command to standard input of another command. So here's an example. Let's say we have a file f1.txt, and we want to get the first 15 lines from it, but we're not saving the output to another file. Instead, we're using the pipe operator. So we're directing this output from this command into the input of this command. We would, so from those first 15 lines, we would select the last line, which would be the 15th line. It's very useful also with file manipulation. The pipe, this is where the pipe operator is usually located on the keyboard. You have to press the shift and this button. Grep, we will use this also in the workshop, allows us to do pattern matching. Basically, we can select lines based on a pattern. Let's say we have a file and we want to select all the lines which contain the string ABC. So we just specify the string and it will print out all the lines, or if we want to, we can save those lines in a output by using the greater than operator. The -v option allows us to not select lines which contain this string. There are a lot more complicated things that we can do with this, but for the sake of time, which I think is running out, I guess, we will not go over them, but I will show you some examples in the workshop. So sed, basically like as in Excel, it's basically search and replace. I'm sure many of you have used Excel and searched for something and replaced for something. We type the command sed, option S. In the first field, we provide the value we want to replace, and in the second field, we want to provide the value that we want to replace the first value with, and this will replace the first instance that it finds let's say the first time it finds the string green, it will replace with blue and exit. If we want to replace all occurrences of green with blue, then we need to add the G at the end. The G means global, so all occurrences of green. So yeah, the bash, I'm saying bash. What does it mean? So it's the command line interface, which allows us to use the commands in the terminal. And also it allows us to, it's like an extension to the commands in the terminal. It allows us to write some scripts. It has syntax, just like other programming languages. It has loops, lists, arrays, and so on. So we can basically do some sort of automation for our data processing. Oh yeah, what is a script? It's a script basically is just a text file that we write with some sort of commands. In this case, all the commands that we're running in the terminal, we can also run through using a script. And the advantages, firstly, that we know we can after we run the commands, we can easily go back and see, let's say, the inputs, the outputs, which commands were used. We can modify the script, run it again. We can make exceptions. We can use the file name globbing in the scripts to run through multiple files with a pattern. Yeah, little bit of syntax. The lines that start with a hashtag, they are considered comments and are not executed. Yeah, so here's an example. We could write all of this in a script, or just as well, we could just write these two commands in the terminal. The script is the more convenient if we're working on a lot of data, and it's also reproducible because we can follow where the data comes from, where it goes, and we have it all written down. Yeah, the variables are defined with the equal sign. There can't be any spaces, which I know that there can be spaces in other programming languages, but here there can't be. And we can access those variables with a $sign. So for example, if we specify that x equals 3 and we want to print out the value of x, we will type echo space $sign x, and it will print out 3 on the terminal. So yeah, everything in single quotes is interpreted as text. The same goes for double quotes. The thing to remember here is that it doesn't matter if you use single quotes or double quotes, but for a single string of characters, you have to use, let's say, single quotes in the beginning and single quote at the end, or double quotes at the beginning and double quotes at the end. You can't mix them. And just a good thing to remember, if you started using single quotes in a script file, it's just good practice to keep using single quotes. It doesn't change anything if you switch to double quotes, but it improves the readability. Command line substitution. So basically, what it means is that when we're writing a script, let's say we want to create a variable called line count, but the value that we want to assign to it is an output from some other command. So by using the $sign and brackets, we can type the first command in these brackets, and the output will be assigned to line count. Let's say we want to count the lines of a Fastq file, or we can use this type of convention. Yep. So loops, for loops. If you write bash scripts for genomics analysis, you most likely will be using a lot of for loops in the beginning. And they are very convenient, allow us to loop through lists of, let's say, file names or other patterns that we matched with the file name globbing. Here's the syntax. Yeah. So there's a variable name that we are creating the variable basically, and we are assigning values from a list to that variable name, and we can do something with that variable name, whatever that might be. So here's an example. Here's a list of numbers, and we create a variable called number. So we are enumerating through the list, and each time for each iteration, it assigns a value to the variable number, and we can, let's say, echo the variable number, and it will print 1, 2, 3, 4 in the terminal. Yeah, the same with, we can also include the substitution. Just we can we don't have to write out the list. We can just use the pattern matching and create a list in that way. So done with the Linux. Moving towards the end of the presentation. So about the workshop. Basically, we will be working in a high-performance computer today, also called a supercomputer, also called a cluster. So basically, most of the time when you're doing some type of genomics analysis, you will connect to a server. First of all, the data is often stored on the server because one, it takes up a lot of space, and two, there's the data privacy protection, so you can't really put it on your personal computer. But also the servers and the HPCs are infinitely times more powerful than your personal computer, so it's just very convenient to do it there. So let's say we log in we connect to the login node, which is just it's a computer which is connected to many other computers, which are a lot more powerful than the login node. And from the login node, we send our tasks to these other computers, also called computational nodes. So yeah, all of these computers are connected with a super high-speed network, and we will be using this system today in the workshop. So here's a typical data analysis workflow. Most often than not, these steps will be repeated. It doesn't matter what you are doing, but if you're receiving raw reads, the first thing what you have to do is check the quality. Then maybe you have to trim to improve the quality, or trim off the adapters that might have been left from sequencing. You want to select, if possible, the highest quality reads. And the next step, which usually follows, is the realignment. And in our case, I've circled, I put this branch in a box because we're going to be doing a variant calling workflow today. So we need to align to the reference genome, call the variants, annotate them using some sort of database, and yeah, then we look for the variants that we want to use. So yeah, the variant calling, or detecting single nucleotide polymorphisms. So basically, it's variation with a single position in a sequence. It occurs very often, and it's been thought that roughly 45 million SNPs are in a person's genome. So yeah, the variants actually, so this is a visualization of a single nucleotide polymorphism. Here we can see, on the bottom, we can see the reference sequence that we provide as an input to the mapping process and the variant calling process. And here we can, the gray lines are reads. They are overlapping, and there are a lot of overlapping reads, which means that we have high coverage, and that helps with reducing the probability that this single nucleotide polymorphism call is by chance, or is it that is false positive. So we can see that in basically all of these reads, the base G has been substituted with the base C. And sometimes these changes can cause serious there can be serious effects on the amino acids that are being coded, and respectively to the proteins that are being made from the amino acids. So yeah, today we'll be looking at a sample of familial hyperholesterolemia. Okay, I can't pronounce that really. But yeah, okay. What's the problem with this? Basically, the people who have this have a presence of high plasma cholesterol levels. And what causes this is a gene alteration inherited from a parent. And as far as I know, there are 4 SNPs that have been associated. There are multiple SNPs, but in 4 genes that have been associated with this disease. It occurs in 1 of every 250 individuals, and the results of having this is early development of heart problems, just like atherosclerosis and coronary heart disease. So this is not a good thing, and we'll have one sample today, and we want to check if in that sample we can find the SNP for one of these genes. Overall suggestions for work in bioinformatics: always read the documentation, read publications, read forums. If you use a program but you don't read the documentation, that's a good way how to mess things up, how to get unreliable results, how to get results that you can't explain, and that's not good practice. The second thing is to think about the design. Here I'm talking about the design of the experiment. So how are the samples prepared? In what batches were they prepared? How were they sequenced? Which chips were used? Because how were the sample variables? How were they distributed across experimental batches? For example, if we have tumor and normal samples, and we have only women in the tumor group and only men in the normal samples, then this sample variable is overlapping with the experiment variable, and we can't really detect the real biological signal. So think about the design. Yeah. So ask questions. I have provided some links in the next slide to forums where you can ask questions. Software is often deposited in a public repository like GitHub, and there you can communicate with the developers, ask them questions. They're the best people to answer them. So communication is key, and by this I mean, if you're a person like me, which mainly just analyze data, then for me it's important to talk to the lab technicians who have prepared the libraries, who have sequenced the samples. I want to know all the batches that samples came in, which samples were sequenced maybe multiple times, because that happens if you get a bad sequencing run. You need to resequence them, and that adds unwanted variation to the data. And I need to know all of this information, as well as information about the patients that provided the samples. Let's say, were they smokers? How old were they? Were they? What was their age? And so on, so on. So all of this information is very useful when you have data and the output is not what you expected, and you're looking for problems. But if you don't have this information, you can keep looking forever, and you can just drive yourself crazy by doing that, because you don't have anything to test to find the reason of these problems in the results. And yeah, the last and maybe the most important is that Google is your friend. If you have a question, most likely somebody else has asked it before, and you can find it by searching on Google. Often the problem is that you need to use the right keywords. And if you are starting out, let's say in this field, that can be a big problem because you don't know all the terminology. Like nobody knows it, of course. I don't know it, but to find the right terminology, to ask the right question, that could be time consuming. And if you don't know the right terminology, then often you can't find the answers to your questions, but it doesn't mean that they are not there. You just have to keep looking and try different ways. Yeah, here are some useful resources. So GitHub software is hosted here. BioStars, that's a forum. You can ask any questions about bioinformatics here. Bioconductor, this is for R and R Studio, basically. They have a forum for questions, but also they are hosting repositories for software developed for the Bioconductor project. So basically, those are packages that you use in R. Bioinformatics Stack Exchange, also can ask questions there. And another forum is Sec Answers. And yeah, there's a meme for the conclusion. When you want to do something, you're really excited, but all you do is run into problems, then you think that bioinformatics got hands. Yeah, so thank you. That's all from me.
Rihards, do you need like a 5 minutes break or you want to start?
The workshop?
Yeah.
When was it supposed to start?
At 11.
Oh, hell. Okay, maybe we should take a 5-minute break because I need to drink some water. I've been talking a lot.
Okay,
yeah.
so 20 past 11, we start the workshop part, yes?
Okay.
Okay. So you ready, Richard?
Yes, maybe.
Okay. Okay, then we are starting the command line workshop.
Okay. Well, I'll share my screen now. I hope that you have all received the PDF document, which we'll be following, and I hope that Laura has sent you the login information for the cluster and your participant number. Okay, so I don't know how many of you tried to set up the Windows subsystem for Linux, or how many of you tried to set up the PuTTY software, but it doesn't matter basically. So we can open the Ubuntu or PuTTY or just the terminal if you are on Mac OS. And the first thing we're going to do is connect to the cluster. So maybe I'll make it bigger. So we're going to type SSH and the username, which is bioms01 at ui-2.hpc.rtu.lv. We press enter and it asks for the password, which I will copy and paste with the right mouse button click. I clicked it, nothing appeared, that's normal, and I press enter and I'm logging onto the cluster. So here I can see that I'm in the user account of bioms01 at this login node. So we can type ls or list, and we can see the contents of the home directory. There's a bunch of stuff that that's not for us, but what we want to do next is create a session, interactive session on a computing cluster. So what we're going to do now is type the command qsub.i, which means interactive -i -l. -l means a list of options, and we're going to specify that we want to use nodes equals 1. That means one computing node, colon ppn equals 2. That means processing cores of that node, and we'll press enter. We see that we're waiting for a job with this number to start on a processing node, which is called Rudens or Autumn in English. So here we can see that our account is now connected to the processing node instead of the login node. Yeah. If we type list again, we see the same thing, because they are connected, and all that this interactive session does means that we're going to be sending all our commands to the computing node, but in reality we are still located at our user account. So now we have to switch the directory to INTEGROMED. We can type list again, and you can see the folders of all the participants that registered, which were 98, I guess. I am participant 99. So find your participant number and change into that directory. For me, it will be participant 99. Oh, also you don't have to type the whole word participant. You can just start typing it and press tab, and it will autocomplete. And the only thing it can't autocomplete is the number, because there are many different numbers and you have to type that in. Press enter, and we can see our location. And we are in this directory. Type list again, and we have a directory called resources. If we change into that directory, and type list, we can see the contents. When we have a index for a reference genome, we have the reference genome, but in this case, it's just for the chromosome 2, because this Fastq file is a subset from this specific chromosome. This is a very small Fastq file. It has like, I think, 28,000 reads or something. So now we'll look at the contents of the Fastq file, which is a common sequencing date format. So we can type less L16 and press tab, enter, and we can see the contents of the file. So basically, each read is made up of 4 lines. The first line is the read identifier. These reads are from the Ion Torrent sequencing platform. For different sequencing platforms, the read identifier can be different. This first one is for the sample. This one most likely is for the lane or the run, and this one is the actual the sequence for the sequence, the identifier. So this is the sequence. They are of differing lengths. In this case, we have a plus sign, which is a sort of interesting artifact from when this file format was created. And in the last line for this first read, we have the quality information coded in ASCII. So basically, these symbols, they have to be translated into actual numbers. But this is the more efficient way how to store the qualities. That's about it for the Fastq file. So now we'll go back one step. As I told you in the presentation, we can type cd ... enter, and we are back in our participant folder. One thing that I didn't mention in the presentation, there's a command called clear. So when you're feeling that there's too much information on the screen, you can just type clear, enter, and it just empties the screen, but the information is not lost. Or we can type list again. We can see that it's still there. So the first thing we're going to do is going to be the read quality evaluation with a program called Fastqc. But before that, we're going to create the results directory and also directory for this Fastqc output. So we're going to do mkdir -p, which allows us to create directories within directories, so directories and subdirectories. And we are going to do results /01 _ Fastqc. I like to follow this naming format with example 01, 02, 03 for each subsequent action I perform. So it's easier to track later if I have to come back and see what was done, and then I can see the order. So that's easier for me. Now that it's created, we can type list, and we can see that there's a directory called results. If we change it into it and type list again, we can see that there's a directory called 01 Fastqc. So the command worked. Now we'll go back to the participant directory and perform the Fastqc for this sample. We're going to type the command name, and now we have to provide the input for this command. And the file itself is located in the resources folder. So we're going to do a relative path to it with resources /. We can just type L16, press tab, and it auto completes because that's the only file in that directory with the beginning L16. So it can easily find it and complete it for you. Next, we're going to specify how many processing threads we're going to use for this job. And that's going to be 2. Incidentally, we already have set up the interactive session to only use 2 cores. So we're not going to exceed that, but you could set more cores, let's say, for this program. But there comes a point where adding more processing cores doesn't speed up the process, because not all programs are optimized equally and not written. Maybe some programs are not optimized at all, and they can't use many processing cores. Some can't use very many. And let's say some programs are optimized to use 16, and I know that more is not necessary. So we're going to use 2. And next, we're gonna specify where we want the output of this to go. So we're going to do results and 01 Fastqc. That's the directory we just created. And now we just press enter. And here's an output. We can see that it tells us how much of the job is done. We can know that it's done now that we can see this information about where we are located again, and we can easily type something and it works. Okay, so now we're going to go to the results, 01 Fastqc, press enter, list, and we can see that we have 2 files. First is a HTML file, which we can open in the browser, and the second one is a zip file. Basically, the zip file contains multiple other files which were used to draw the graphs for the HTML report. So we can't open the HTML report in here. So I'll ask you to open the WinSCP program, which I also asked you to install. So here we have to again type the information for the login node, which is ui2.hpc.rtu.lv. Port number is the same. Username is bioms01, and the password we can just copy it and press login. So on the right side, we can see the information on the server, and on the left side, we can see let's say if you're on Windows, we can see our Windows folder system. So if you need to, you can create, let's say, a folder on desktop or somewhere else where you're comfortable, where you're going to store some of these results. So we can just click through these, find our participant number, results, Fastqc, which we just created, and it's a simple drag and drop interface. So we have just moved the HTML report from our server to our personal computer. So now we have to open a browser, and we can simply drag the HTML report in the browser. So now we'll go through the Fastqc output. So the first thing we see is a chapter called Basic Statistics, and here we have a file name, file type, encoding. So the sequence quality information in the ASCII format, it can have different versions depending on the sequencing platform which was used. So this detected that the version is compatible with Sanger or Illumina 1.9. You can see that we have 27,000 sequences approximately. Sequences flag disprove quality 0. A sequence length. This shows us that the shortest sequence we have is 8 base pairs, and the longest is 293. And the average GC concentration in our reads is 51%. So this sequencing sequence length can also differ depending on the sequencing platform that was used. I have to also mention that these are single-end reads. Nowadays, at least for me, paired end reads are more common, but sometimes it's better to use single-end reads. Okay, so here we can see a graph of the read quality. This is an aggregation. So basically on the y-axis, we can see the thread quality scores, which are calculated from the ASCII symbols that we saw in the Fastq file. These three zones specify the goodness of reads, if you could say so. So the green zone is where we want to be. The orange zone is somewhere in between, not great, not terrible, and the red zone is where we don't want to be. So these whisker these are whisker box plots. Basically, they show the first to the third quantile. The red line is the median value. I think this is the top whisker is the 90th percentile. Bottom whisker is the 10th percentile. The blue line is the average value. So yeah, basically, it's very common that you have a lower quality reads, lower quality bases at the end of the reads, as we can see here. The quality drops quite dramatically. Also, the longer the reads, the bigger the possibility that the quality will drop due to sequencing errors. That's due to the chemical reactions, which can, I guess, the reagents can go bad during the sequencing process, or there can be adapter contamination, which also causes lower sequencing quality. You can also see on the x-axis the position and read in base pairs, and first we have 1 through 9, and then we see these aggregate batches of positions. That's something to take in to keep in mind. So on the next chapter, we can see per sequence quality scores. So on the x-axis is the mean sequencing quality. So when we're looking at this graph, we want it to be as sharp as possible. We want more sequences to have the same, and it's ideal if all of our sequences have the same mean sequence quality, and it's more to the right side, where which means that they have the higher sequencing quality. Here we see that it's not quite the case. We have some sequences with very low sequencing quality, and most likely we will have to trim these off because they will, when we will try to align the reads, they will cause spurious alignments, and they won't align anyway. So there is no reason for us to keep them. And on the y-axis, we can see the number of reads which have this quality. So, for example, we have about 4,500 reads with a mean quality of 28, which is good. Actually, this quality for ion tolerant sequencing platform is quite good. From what I've seen, it's usually worse. The quality is usually better on the Illumina or MGI sequencing platforms. So the next chapter is per base sequence content. Ideally, if we have a library prepared from random fragments, we would like to see all of these lines which represent bases. We would like them to be parallel to each other and consistent, so straight in this case. We can see that that's not really happening here, but it's also nothing really too bad. What we really don't want to see is this big difference, which coincides with the quality plot which we saw above. That this right here means that there's at this position there's a lot of G bases, and very little amount of A bases, and this is quite a big difference. I think 40% this is, and usually when there's such a big difference, the software will report that there is an error or that we have failed this quality check. Also, this plot might be useful, or it might not be useful depending on the type of samples you're processing. It can differ very much for, let's say, RNA sequencing or 16S ribosomal sequencing, where there are a lot of repeating bases for the not variable regions. So you also have to take in mind that if you're failing this quality check, it doesn't mean that everything is bad. You just have to look at it in the context of the data that you are processing. The next chapter is per sequence GC content. As I understand that when our samples are amplified, there's a bias in the PCR reaction where it likes to amplify the sequences which contain more G or C bases. And we don't want that, because that introduces, as I said, bias in our data and results in not optimal results. So yeah, basically when we this is the blue line is the theoretical distribution. That's what we like to see ideally, but our real situation is these spikes, and well, in this case, we know that this sample is a subsample of a sample, and we can't know how the subsample was created and how well the subsample represents the actual sample. So it's normal that we have differing, just because we have differing read lengths, we have these spikes in this GC distribution graph. But let's say we would have this bell-shaped, we would have something very similar to the bell-shaped curve, but there would be a one very, let's say, a large spike to the left and not these little spikes. Well, that would mean that we have some kind of contamination in our sample, most likely from a different organism. So that's something to watch out for. The next chapter is per base N content. So the sequencing platform, when it can't determine which base was in the read, it assigns it a value of N, or like not known, basically. We can see here that we have no bases with or very little bases with the N values, which is a good thing. This can occur from data with low quality, and then we know that we have problems with the library preparation process, or these can also occur from adapter sequences which are still left in the reads, and they often have lower quality, and therefore can't the platform can't assign it a base. Well, this is fine. And here we can see the sequence length distribution. As we saw in the summary chapter, that the shortest sequence was 8 base pairs, and the longest was somewhere around 290, and that's a very big difference. Typically, let's say we would have a paired end library with the length of 2 times 150 base pairs. So we would have a graph similar to this, when we would have a spike, let's say at the 150 mark, and that's most likely the ideal result. We would have very little sequences with a shorter length, and most likely no sequences with longer length. But this is this is due to the subsampling process to create this file. So we will remove some of these. Typically, if this is not caused to avoid, let's say, adapter dimers, we will trim off like the first 35 or 30 or 40 bases from these reads. Yeah, just to avoid the contamination of the adapters which were sequenced. Yeah, so the sequence duplication level. So basically, if we have a random, we have a library from random fragments from the sample. Ideally, if all the reads were represented, we would not have many sequence duplicates. So duplicates means that we have many reads which start and end at exactly the same position. If we look at their by position, I mean if we would compare them to the reference genome, like we would map them against reference genome, and they would start in exactly the same position and end in exactly the same position, and that is not an ideal variant. So yeah. Typically, I see this spike around 10, and which that is not bad actually. I mean, we have like 40% of the sequence with duplication level of 10, but after trimming, this should lower, but this is not bad. Also, in this graph, you also have to take into account the sample type from which the libraries were prepared, as I mentioned before, for RNA sequencing or for 16S sequencing, or basically for samples which already have small genomic diversity. The duplication levels could be really high, but that doesn't mean that your that the sequencing wasn't successful or that there's a problem with the libraries. That's just like the characteristics of the data you're analyzing. So here we see the overrepresented sequences or sequences that are so specific. These are called K-mers or short sequences that appear in our data, but can't be assigned as reads. These could be either adapters, adapter dimers, or some other contamination. But we can see that there's not a lot of them, and after the trimming process, there will be even less. Yeah, on this last column, there's a possible source. That means that the software checks against its database whether it can define where the sequence has come from. Basically, it checks against the most popular adapters, I think. But here we have no hit in all the entries. Yeah, on the adapter content, we can basically see that it has detected that our data has some solid small RNA adapters, but that's a very little amount, or basically zero. So I would say no adapter trimming necessary. For the trimming software, you can either trim just by the read quality, or you can provide adapter sequences that you want to look for in your reads and trim, or you can do both. Or you can do none if your reads are really good quality. So yeah, this will be it for the Fastqc. Now we'll return to the terminal. I'll go back to the participant directory, clear the screen. The next thing, okay, if you look in the PDF file, actually I have put in an image of the it's like that's an example table of the Phred score. So if for a base we have a Phred score of 40, that means that the possibility that the base was called incorrectly is 0.01%, or the accuracy is 99.99%, and that's really high. For 40, that's a really high value. Usually we don't see those. Like the good quality reads are 30 plus minus, maybe 30 to 35. Nowadays we can see those very often. maybe 20 could be the cutoff at which we would throw out the sequences which are under 20, because 1% it doesn't seem like a lot if you look in the context of let's say 100 bases, but if you look in the context of genome with 3 billion bases, the 1% does add up. Yeah, but also the cutoff criteria depends on the overall quality of your data. If your data is low quality, you don't want to trim it. Let's say the mean quality is 18 or mean quality is 20 or something. You don't want to trim it at 20. So in that case, you'll just throw out most of your sequences. So most of the things we do here, you have to look at the context of your current situation. Next, we'll perform sequence trimming, but first we'll create the results directory again. mkid results 02 Fastq. Fastq is the name of the software that we're going to be using for sequence trimming. And next we're just going to call the command fastq -w2 means use 2 processing cores. Cut front. You can just copy. You don't have to write. You can also copy paste. Just copy from your PDF and right click in the terminal, and you don't have to write it by hand. Cut front. So cut tail. Cut window size 4. Cut mean quality 15 for this case. -L 35 means we'll throw out any sequences that are shorter than 35 base pairs, and that E15 means throw out any sequences which have mean quality lower than 15. And then we'll provide the input with -I, and we'll go resources L16, use tab for completion, and then -O, we'll go results 02 SPs, tab for completion, and we'll just name the file L16.trim.fq. fq is just short for Fastq. You can use Fastq or fq, it doesn't matter. Some of the options here: cut front, cut tail, and cut window size basically means just that the software, if you imagine a read, then it's this means that we're trimming by quality. And basically, it's sliding like a window of 4 bases from the front and from the back, and checking whether those bases have mean quality 15 or lower. So if it's lower than 15, then those bases are removed. Just press enter here, and there's some output from the software. We can see that 86% of our bases are with the quality Fred score 20, 44% with the Fred score of 30. We can go to the directory, and we can see that we have the trimmed file. Yeah. So I'll just go back to the participant folder, and now we're going to rerun the Fastqc. It's a good practice to rerun the quality check software every time you change something about the read. So whether it's quality trimming, adapter trimming, removing ribosomal sequences in the case of RNA sequencing, every time you do something like this, it's a good thing to rerun the quality check software to see if you haven't cut off too much. How does the quality change? If it changes at all, maybe your thresholds need to be more strict and stuff like that. So we'll just do Fastqc again, but this time as input, we'll type results Fastq, and we'll input the trimmed sequencing file. -T2 for 2 processing cores, and -O for our output. No, I guess no. First, I guess I forgot to create a directory. Yeah. First, we should have results 03 Fastqc trim. Yes, and now we're gonna run the Fastqc again. So we can see that the file that we processed is the trimmed file. Yeah, so again, we'll go to the WinSCP software. We can refresh, and we see that we have new directories which we just created, and new reports with the trim in the file name. We can just drag and drop it again, open the browser, and drop it in the browser. So now we can see that we have lost about 3,000 sequences from the trimming. The shortest sequence is now 35 base pairs, as it should be, because we specified that we don't want any sequences shorter than 35. In this graph, we already can see that the end of the reads have increased quality from before. I'll just go back, and we can see that it drops quite significantly, but here it's more stable. So the per sequence quality score graph has also improved. It's sharper than before, but that's also due to the fact that we did the quality trimming at the mean quality of 15. So that's perfectly sensible. Per base sequence content, we're still failing this, especially at the end of the reads, where we have a very huge difference of C and G base. The GC content plot hasn't improved, but most likely for this specific sample, we can't improve it due to the nature of how it was created, that it's a random subset of sequences. So basically, there's most likely no way that it will represent an actual GC content of a sample. Per base and content is still 0, which is great. Sequence length distribution, the graph is still bad, but we can see that our sequence length starts from the 35 bases, which we specified. So actually, no, the main reason for this was because very short sequences have poor mapping quality. Duplication levels haven't changed, still overrepresented sequences, and no adapter content. So some things have improved, some things haven't, and yeah, but in this case we have to understand that we are working with a specific sample, which is not maybe the greatest, but it's what we have. Okay, so next we have a chapter on downloading the human reference genome. So I won't be doing that. I have already done it, and I have prepared the reference genome index and copied into your participant directories. But basically, there are multiple sources of reference genomes. For example, UCSC, Refsec, or Ensemble. In this case, I used Ensemble because from the information I've gathered online, I guess it has the most comprehensive annotation. It's possible that different reference sequence sources could be better for different analysis, but that kind of information you'll have to find out on your own by doing the research. Yeah, so next chapter is to build the index for reference genome. So I've done that. It's in the research folder. And next we move on to the sequence alignment. So first we'll create the results directory, which we'll name the same as the software, with which will be performing the alignment. Okay, and next the command is HiSet2. First, we specify the name and location of summary file of the mapping. So I'll type in the chromosome just for, let's say, if I'd had to come back later, I would instantly see that this sample was mapped exclusively to the second chromosome. -X, we provide the reference index. And here we don't have, because the reference index is made from multiple types, multiple files in this case. Also, the reference index is different for different alignment software, and they can't be used interchangeably. So do be very careful about these indexes. Read the manuals, read the documentation of how to create them and what they mean. So here we just have to provide the base name without the file extension. Next, we specify the processing code, which is 2 again. -U will provide the input to the software, which will be the trimmed reads. And -S will specify the output in the folder which we just created, and we'll name it L16 chromosome2.SAM. So SAM is a sequence alignment map. I will talk about the format next. So already here we can see that only 39.74 of our sequences aligned to the reference genome, which is not a lot, but I'll say that most likely is due to the fact that we have a lot of sequences with different lengths. Typically, the longer the sequence, the bigger chance that it will align correctly to the reference genome. The shorter the sequence, the bigger the chance that it could map to several locations in the genome. But those are called multi-mappers, and we don't want those in our data. We can see that 0.1% of our sequences aligned more than one time. Those are the multi-mappers. 39% aligned exactly one time, and 60% aligned 0 times. So now we'll go to the output folder, and here we have the summary file. Yeah, it's the same as we just saw on the terminal, and we can also open the sequence alignment map. Okay. Here we'll use the less command, but with the capital S option, which, as I told you in the presentation, that it disables the line wrapping basically, so the file becomes easier to read. So here we have the header section, the first 3 rows. We can see that the information is unsorted, and we'll have to sort it next. And here's the actual body of the file. So each entry is for a sequence. The value of 4 here means that it didn't align. So, and therefore we also see no values in the rest of the fields. Why are there so many 4s? Well, it makes sense because we just saw that 60% of our sequences didn't align. So it makes sense that there are a lot of these 4s. Yes, so next we'll move to the sorting. We'll convert this SAM file to BAM file. So basically, sequence alignment map. That's let's say a human readable format, as we just opened it and we could read it, but we'll convert it to binary alignment map. So what it does and why are we doing it is because it saves space. Usually when we align samples of the more normal size, the SAM file size could be like 40 gigabytes. And when we have like 100 samples, that adds up to a lot of space on our storage. So we want to convert to the BOM files. And also a lot of variant calling and also and other downstream programs require that the reads that were mapped are sorted by coordinates, in this case, the chromosomes or scaffolds, because we have only chromosome 2. So we'll go back to the participant directory. I'll clear the screen list, and we're going to do sum tool sort -@2. So we're specifying again the processor threads that we're going to be using. So here it's a little different. First, we'll specify the output. Yep, and we'll use the same output directory as for the alignment. And this time I'm adding the word sorted to the file name so that we know that it has been sorted. So bam, and now at the end, we'll provide the input file, which ends with SAM. So it's done already. We can go to the directory and we see that we have a BAM file. So I can't open it with less because it's binary. I can't read it anyway. So a way to read it is to use sum tools view and just provide the file as input. Then actually, yeah, the pipe operator. Now we can test the pipe operator that I was telling about. We can select the first 20 lines and pipe it again to less. So here's the binary alignment map, but in yeah, we are reading it. So here we can see again the specific tags of whether it mapped, it didn't map, or how it mapped, the coordinates, and here we can see a cigar string, which is a sort of interesting name, but basically all it does, it characterizes the alignment for this sequence. So basically 79M means that 79 bases matched, the first 79 base matched. Then there's one base with an insert, so our sequence had one base that the reference genome didn't. 22 again matched, one deletion, so we didn't have one base that was in the reference genome, and again 10 bases matched. So next we need to index the BAM file. So basically indexing both for the reference genome or for the BAM file, it allows other software to access the file more efficiently and faster. The index itself, you could think of it like it's a map basically. So the software could look at the map and it could easily locate what it was looking for. It doesn't have to read through the whole file. It doesn't have to test against every sequence to find what it is looking for. So we'll need to index it now. I'll go back to the participant and we'll do sum tools index -@2, again 2 processing cores, and we just need to provide the input, which is again the same folder, and we'll provide the sorted BAM file. So quickly let's check, and we could see that we have a BAM file and we have a BAM by file, and the by means that it has been indexed. Okay, so let's move back to the participant, and yeah, so we're nearing the end, and I think we are actually also running out of the time, so I'm trying to move quickly, maybe too quick, but we only have as much time as we do. So yeah, let's create a new results directory for the variant or SMP calling. So basically we want to find locations in our sample data which have different bases from the reference genome. In this specific case, we will only be looking for polymorphisms or base substitutions. The software that I'll be using now is also capable of calling insertions and deletions, but today that's not what we are looking for, basically. So first, we'll call the variants with some tools and pile up -uf, and we need to provide it with a reference genome. So I just typed homo and pressed tab and it auto-completed. Next, we need to provide it with the sorted BOM file. So we actually don't need to provide it with the index, because when it will try to look up the BOM file, it will see that there's also an index with the same file name prefix as the BOM file. So it will find it on its own. So now we'll pipe the sum tools and pile up output into PCF tools. And the option here is call for call variants, and specify the algorithm. Specify that we only want the variants. And we want to skip variants that are in those. And finally, we'll use the greater than sign to redirect our output to our created directory. And here I can use, so you can see that this line is getting very long, and I can use the / operator to transfer this to the next line. Basically, it will be the same command, but I will not run out of the space. You can also use this in the scripts. So here, just type results 05, and the file name will be chromosome 2, RedCF, tools, smps.wcf, and this WCF, that's a variant call format. This is very common file format for variant calling, but it is also used in other types of analysis. It's quite universal. So I'll just press enter. And it's done. We can go to results 05. We can see that we have the VCF file. And we can open it again with -s to check the contents. So, okay, this is the variant call format file. And first we have a header. Basically, for this file, most of the header consists of explanation of what kind of information you can find in the info field. So, as I mentioned before, the files are not always consistent with the delimiters. So here we have, let's say, 7 columns delimited with tab, but we have the column info, and the values in this column are separated by semicolons, or sometimes it can be pipes or whatever. So, okay, on this, these are the actual the SMPs we called. Yeah, so here are the position in the reference genome. First, we have empty. We need to add information from a database to get the SMP ID, which usually starts with an RS and followed by numbers. Here we can see the reference allele in the reference genome, and here we can see the allele in our sample data, which is different from the reference. That's why it was called. The mapping quality in Phred score, and information about the variants. So maybe now we want to, okay, we can see this file, but we don't know how many SMPs were called. So here we'll try out the Grep, which I mentioned in the previously, basically. So we can do this. So we want to exclude the header and count how many lines are left after excluding the header. So we can grep grep dash v, which means that exclude lines that contain these symbols, and we can do double hash because a lot of the header lines contain double hash, or also with the pipe we can specify another pattern, which is a single hash, and single hash was actually for the column names, but for this specific reason we're not interested in the column names as well. And here we'll provide the file as an input. And next we just pipe the output of the grep command into input of the wc-l, which will count the lines. So here we can see that we have 34 SMPs called. Okay, so next we are interested in the disease which in the variants that are occurring in the and causing the disease which I mentioned before with a difficult name. So what we need to do now is add annotation from a database so we can determine where is the variant, in which genes are the called variants located, what are the consequences of these variants or the base changes basically, and we will be able to filter out the SMPs which have, let's say, moderate to serious consequences. So now I'll go back to the participant directory. Yeah, in the PDF file you can see an example of the VCF file header and what information is available there. So next we have the SMP or variant annotation chapter, but here I have shown you how to determine which databases are available and how to download them, but I have already done that for you. So next all we have to do is just annotate our called SMPs. So this will be a little different than before because this software is written in Java. So here we can't make an alias for it. So we don't have to write the full path to it, but that's all right. We can do it one time. That's nothing terrible. We can also use the autocomplete and just go to software, SMPF, /SMPF and .jar. .jar means that the files, the program was written in jar. Now we specify the database version that we're going to be using. So the assembly is basically the latest version is the 38, but the 99th is not the latest subversion, but it was the latest subversion which was available for this software at this time. So next we have to provide the input, which will be in the directory which we created recently, 05 SMP call, and then with the greater than sign we will specify the output folder, which will be the same, but we'll modify the file name so we know that it has been annotated. I'll add the N at the end. So it will annotate the called variants now. Oh, and yeah, about this option. So basically this means that I'm specifying for this command to use 4 GB of RAM. Some programs are very RAM intensive and you just need to provide it more RAM to them, or otherwise your computer might, or your process, first of all, your process will end because it didn't have enough RAM, or if you actually use all of the RAM available and it still needs more, your computer might freeze and you'll have to restart it. But for this purpose, 4 GB will be sufficient. But this process is taking some time because the database is very extensive. Oh, I'm currently reading a message in the chat. So one person had trouble logging in to the server. He's actually using a Linux distribution, but it didn't work. I guess my first question would be, what is the error message? Okay. Oh, so okay, yeah. So basically, that's not an error message. Your computer is recognizing that it doesn't know the cluster computer, and basically it's considering that this might be a security like breach or a threat. Or it's asking you if you want to continue your connection. Basically, you would have to type yes, press enter, and then you would be able to enter your password. When hitting yes, nothing happens. That's weird. I'm pretty sure that it should work when you type in yes and press enter. It's actually asking you if you want to continue connecting. Were there any other errors after you typed yes? Actually, this process is taking quite a long time. That's maybe because we are all trying to do it at the same time. Now I'm reading the message from this one person which couldn't connect. Yeah, this is very interesting actually. In theory, it shouldn't matter which distribution you're using. So unfortunately, I think we have like maybe 5 minutes left or something. So we'll not be able to fix this problem right now, but it's worth looking into it later. This is taking surprisingly long. a surprisingly long time. When I tried it previously, I think it was pretty fast. Laura, are you here?
Yes.
How much time do we have left? 5 minutes?
Yeah, 5 minutes, yes.
Okay.
Maybe leave the login active for a week, and I will send this your demonstration of the workshop, and if someone wants to fulfill it in a week, then they can.
Yeah, I guess.
How much time do you still need? But
okay, we actually were at the end, but this is basically the last command, but it's taking a very long time for whatever reason.
you think it's something with the server, or
Did I mistype something, or there's just because the load, maybe something is doing on the computational node. Some can't really be sure. I mean, okay, even if we don't get the SNPs annotated, that's fine. We'll just wait those last couple of minutes, but I can just talk you through the PDF file. So essentially, we would get another VCF file, but this time there would be added information in the info field. The thing that we are most interested in is the I guess you can call it the consequences section. And we are specifically interested in missense variants or stop gained variants. So that means basically there's a when this polymorphism happens, there's a change in the amino acid. There's actually a different amino acid is produced. What we will do next, because the VCF format is, it's fine if you want to filter it and you want to subset your SNPs for further analysis, but it's not very visually pleasing, or maybe not very good for presentation. So what we would do next, we would go to the Ensemble website, and they have a software called Variant Effect Predictor, and they have a web interface. Okay, I actually have results from the previous jobs. I can show something, I guess. I tried it before with the same results. We'll see. Yeah, okay, so after filtering for the missense SNPs, we had, how many are there? 8 SNPs left, I guess. And we can see that 3 of them are in the ApoB gene, which was one of the genes in which the polymorphisms cause the disease I mentioned before. So basically, and we have 2 missense variants. Here we can click on the SNP identifier, starting with RS. I can just open it in the new tab. Yeah, so here we have a lot more information about this specific SNP, like genes and regulation that it's associated with. What are the allele frequencies in differing populations? Which phenotypes is it associated to? We can see the 3D protein model, citations, like in which publications it has been mentioned in. Yeah, so we can see, for example, a list of publications. So yeah, we have basically, I guess we have kind of confirmed that this person from which the sample was produced have this polymorphism, but the problem here is that we only have one sample. Ideally, we would have multiple samples to cross-reference, because a result from one sample, there's a probability that it's by chance. Even though we have read coverage and good mapping quality, it's still better to use samples from multiple people, but with the same phenotype of the disease, for example, to improve the possibility that detected the polymorphism is actually there. And I guess we're out of time. I'll check. It looks like the process is still running. That's okay. We can just cancel it by not. We can just press a shortcut combination of Control-C in the terminal. That will cancel the process. And in order to quit our terminals on the computing nodes, we just have to type exit, and now we see that we are back at the login node UI2. And to quit the server itself, we can just type exit, and you can see that the connection is closed, and you can close the SCP. Click yes here, and that's it. You're logged out and good to go. Okay, so I really hope that some of this was useful. It was not meant to be a comprehensive tutorial on how to do variant calling or how to work in Linux, but you can think of it as a primer, an introduction to give you an idea where to look, what to do, how commands work, how software works, and so on. So thank you for your time.
Thank you very much, Rihards. It was yeah, it gave like a taste in how it is to work with command lines and how to, I think about them together with presentation. Very interesting. So yes, then we have now an hour for a lunch break, and then we will be back with lectures from Weizmann Institute of Science about new state-of-art method for analyzing metagenome data. And after that, it's going to be a bioinformatician lecture, Alexander from Lund University, about DNA methylation. And then we will finish with a presentation about metabolomics, and it's going to include sample preparation and also some aspects of that data analysis. So thank you, and now we have an hour break.